Good evening and welcome to tonight's lecture. I'm George Kraw. This evening we'll be hearing about, we'll be learning Earth's biodiversity via space and eDNA. Presenting our speaker is world-renowned biologist Beth Shapiro. Beth, welcome back, and I turn it over to you.
Thanks. I'm going to have to grab the mic from you, I think.
Thank you. All right. Thanks, everybody, for coming out this evening. I don't know what world-renowned means, though. I'm a little bit taken aback by that. Hopefully it's a good thing and not a bad thing. I'm just kidding. It's all good. I appreciate that. Tonight, we are very fortunate that we get to hear from my friend and colleague, Dr. Rachel Meyer. Rachel, I have known for a long time. I'm like to say that I am responsible for her being at UC Santa Cruz. She's joined our department, Ecology and Evolutionary Biology. When? I can't even remember now.
2019.
2019.
Yeah.
But I met Rachel first when she was still at UCLA. She was working with our shared academic mentor, Bob Wayne, who passed away from pancreatic cancer a few years ago. And together with Bob, Rachel and I developed a program that became a course, that became a different program, that became all sorts of things. But without Rachel, it wouldn't be the screaming success, international success that this program is right now. It started off as the California Conservation Genomics Consortium, which eventually got $10 million from the state of California to invest in developing conservation resources for animals. But we had the idea that we really wanted to expand this beyond what we could learn by looking at individual animals. And Rachel then came out and spearheaded this environmental DNA program that we call CALeDNA, California's Environmental DNA Program. And Rachel's going to tell you all about the things that she has done with CALeDNA since the beginning. But I also wanted to point out that it isn't just an academic pursuit, that we're here in Silicon Valley celebrating the relationship between Santa Cruz and the Bay Area. And Rachel is actually a serial entrepreneur. She has had multiple companies, including one called Shoots and Roots, or is it Roots and Shoots? Shoots and Roots, which if anybody tried the cocktail that she brought, I mean, what kind of speaker brings a cocktail, right? I mean, she's winning, right? Then it's kind of the recipes that she's come to use through her knowledge of ethnobotany and the way that people interact with the landscape and how we can use the plants that are around us to more fully enrich our lives and our drinks. So that was the first one, and I'm sure she's going to tell us about the second one. So Rachel, without any further ado, please come up and tell us about the cool stuff that you are doing at UC Santa Cruz. Thanks.
Thank you. You actually made me cry a little bit, Beth. So much time has gone by, and we've put in so much blood, sweat, and tears into growing these programs and seeing some people that are really a huge part of the work that we're doing in the room here, and also seeing so many new faces. I'm really grateful that you're here tonight to dive into some biodiversity learning. And I'm really obsessed with ink painting these days, so when I can, I throw them in. They're not mine. They're stolen from public media. Okay, so first we're going to start with a breakdown of what is biodiversity. Biodiversity is not a term that has been around for very long. It was coined in 1993, and it's from the Convention on Biological Diversity, and it has some pros and some cons. It's complicated, and people often think they don't really understand what biodiversity means. If I say nature, or I say species, or I say native flora, that might connect more to you than the word biodiversity. And I just want you to think about if you are very familiar with the term, or if you already have feelings about if it's a scientific term or not. And then I'll read you this definition, so we kind of sit with it. So the official definition is the variability among living organisms from all sources, including inter alia, terrestrial, marine, and other aquatic ecosystems, and the ecological complexes of which they are a part. This includes diversity within species, between species, and of ecosystems. So it's kind of bulky, right? This is a very large term for the biotic components of nature. and all of the ways that they are shaped and built from within a species to communities. And why should we value biodiversity? Well, of course, humans rely on biodiversity to live and thrive. We're drinking biodiversity and eating biodiversity, and we like having parks nearby, and we build our houses and clothes, and everything is about living things that we depend on, right? But we also can just admire it for its existence, that it exists in the first place. It's 4.5 billion years in the making, and there are over 1 million species right now that are under threat, with humans being the largest threat because of our land use. And so the UN recognized that biodiversity was being threatened and needed protection, and set out for an entire decade of biodiversity with 20 Aichi targets to meet that we would need government coordination on. And after a huge effort, recognizing that if we fail these, we're at loss of exacerbating climate change, controlling climate change or limiting temperature increases is somewhere on here. But if we increase climate change pressures with an increase in global warming to 3 degrees, we're going to be losing 41% of mammals. And just to put this into context, most of the world used to be mammals that were not human or our livestock. And now only 4% of the body mass of mammals is wild mammals, 60% is livestock, and 36% is human. So we're at risk. That little 4% is what's really going to suffer. And already we're witnessing this major effect in the loss of insects. People call this the insect apocalypse right now. There's estimated 75% loss in insects happening. And we sometimes hear about this as the windshield phenomenon. Can you raise your hand if you've heard of the windshield phenomenon? A couple people. This is where you remember driving in the 80s when you had to, on a road trip, pull over every couple hours to clean your windshield because of all the bugs. And now it's not that cars became more aerodynamic, there's just not that many bugs. And insectivorous birds are reacting to this more than other birds. And so we're going to be seeing these cascades of losses of biodiversity and ecosystem services that are provided from insects, from nutrient cycling to pollination to being bird food. So anyways, we set these Aichi targets, and unsurprisingly, we failed. I think only 3 of them were met. And some of the reasons, well, there's many reasons that they failed, but some of the reasons that really resonate with me are poor data collection and lack of uniform accounting tools for biodiversity services. So we can't even estimate the value of biodiversity, and that's part of why we're not investing in or actually making actions to decide to protect biodiversity. You can read some of the other ones on your own. And so, yeah, it doesn't work to say we're going to make all these sacrifices to our livelihoods to help protect biodiversity when it's not already interwoven with our own economy, right? We need it to be good for us and good for the planet. So we need to have a green economy. And there's many challenges to making a green economy work. We have policies that create incentives for conservation that are failing right now. And that's partially because biodiversity or carbon offsets credits are often associated with greenwashing, and sometimes even when they're not intentionally greenwashing, they're ineffective. We also have most conservation work that's not data-driven, and sometimes that's okay, but it's really easy to lead to bad planning, and it's hard to future-proof when you're not having data to support you. And the restoration work, even when we try to do restoration, globally 40% of restoration projects fail. And even when restoration does work, they still fail to meet the level of diversity, of biodiversity metrics that reference sites have. And this is just a few of the challenges to making a green economy. So I want to go back to the topic of poor data collection. This is an image from yesterday from the Global Biodiversity Information Facility, GBIF. This is, red is high, sorry, I left the legend out. Red is high, yellow is low, and black is missing data or data deficiency. And this is a global map of biodiversity observations in the terms of specimens, photographs, audio, every way that we can collect observations of some species existing. And there are almost 4 billion of them. It's tremendous, contributed regularly from over 120,000 data sources around the world. So what do you see here? Maybe you don't see even distribution. Maybe Europe is a little bit off the charts. South Africa as well. So there are some gaps, but we are able to contribute gobs of data, and we have a legacy of data that we are standing upon. Let's not forget that. And so let's spend a little bit of time reviewing, and I'm going to include art as often as I can. This is my friend Meyer Edelman, who did a lot of the artwork for CALeDNA that you'll see around the slides later on. Let's talk about how we measure biodiversity. What are all the tools in our toolbox right now? So I'm not going to go into more deep time biodiversity concepts, but we have specimens for the last 500 years that people have collected that are in herbaria and museums in lots of different types of collections. And these are continuing to be unearthed. This was one that was recently found at Lehman College in the Boogie Down Bronx, where I went to grad school. So we have those types of collections. We also have a lot of technology that helps us put cameras out that can, I don't know if this is actually going to work with this clicker. Nope. Well, these are live meters of hummingbirds, of deep sea expeditions, of audio. You can also go out on your own and go on a safari and go take photographs and make observations and take videos. So we have lots of ways of digitizing biodiversity. And then for the microbial world, we can take soil samples, culture them, isolate those cultures, characterize them, and these are fungal culture collections. So there's a lot of potential out there. Then there's everything that's airborne and spaceborne, with tremendous development in new tech right now. We've got planes that are collecting data. We've got drones collecting data. We've got the International Space Station has sensors that used to face out towards other stars and galaxies, and now they're pointing towards Earth and realizing that they can collect tremendous amounts of information. And we've got satellites that are being dedicated to measuring ecosystem stress, like ecostress, and coordination among also, adversaries around the planet are coordinating, and they're friendly on this type of global data on what's happening to our planet. So some of the biodiversity metrics that these and other types of data can contribute are, I'm going to walk you through right now. So we have richness and evenness. These are components of alpha diversity. So if you look at that corner, that cluster of trees, there are 3 trees. So the richness would be 3 for trees. And then there's another cluster of trees that's also 3. So they would have the same richness, and they have 1 of each, so 33% of each tree, so that's evenness is really high. Beta diversity is looking at the compositional differences. So between clusters, how many of those are shared species? There's only 1 tree that's the same in those 2 closest to clusters of trees, so it would be 33% would be the beta diversity. So there's pretty high turnover. And then gamma diversity is sort of more of the entire ecosystem as a whole. It's alpha times beta diversity. So these are some of the common metrics that we use, and you can calculate them from various ways, from space observations, from iNaturalist photographs, from any data that you have. There was a satellite that was set to launch in 2027. It had been delayed a couple times from previous administrations. It's been put on pause with this administration. It's not going away. It's going to happen at some point. It's called the Surface Biology and Geology Mission from NASA, and it's going to be providing hyperspectral data of 400 different layers for ecosystems, agriculture, coastal zones, snow and ice, and minerals. And you're going to get all of this information that can tell you the brand of car paint. It can tell you so much about what the Earth is experiencing, what the the phenology and the chemical expressions of plants are, for example. And so this is a tremendous opportunity to study biodiversity, but we don't even know the potential of what these data can tell us fully. So, but we know that it's going to happen, and that already this hyperspectral data is available from airborne data and drone data, microsatellites I keep hearing about. So that's another aspect of biodiversity measurement that might be able to be quite large. And then we have, I'm not going to go through all of them, but I'm going to go through one more, DNA. DNA from organisms. There's a lot of organisms that have been sequenced so far. For having some DNA sequences from the 80s when we started sequencing species, having at least a little bit of DNA in a public database, there's 205,000 species. But for reference genomes, we have a long ways to go. Very few species have been sequenced, even though massive efforts have been increasing over the last couple of years, like the Earth Biogenome Project, that's saying, hey, for what it costs to make the human genome, 10 years, $10 billion, $10 billion in 10 years, we can actually give a reference genome to every eukaryotic species on the planet. So it's really cost effective now to generate this data, and it can tell us how different things are and actually help us understand new delineations for species and for groups that are related to each other. And there are other California-based efforts to sequence reference genomes and sequence populations, leveraging that reference genome for high-quality mapping of the genetic data, like the California Conservation Genomics Project, and also more decentralized and sort of local community engagement types of efforts that are not state efforts, like Wise Ancestors. I have to shout out to them because I love what they're doing. These are sort of crowd-funded genomic initiatives. And we can do a lot here for wild species, but also we depend on a lot of the biodiversity sequencing for our foods, our medicines, our building materials, all of the economic species. So as a botanist, I have to talk a little bit about plants. And my start was, I actually got my PhD in eggplant, in Asian eggplant, in medicinal Asian eggplant. And there's a lot of diversity of land races, and they all have different uses and different zones that you can grow them in, different climates. And how many of you farm, garden? And so you're well familiar with that not everything is automatically going to grow everywhere, right? You've suffered. I've suffered. I've had to try to grow all of these in the same place. It's difficult. So after I did my PhD on eggplant, I studied African rice diversity, and I was looking at landrace diversity in African rice, Oryza glaberrima, that was naturally salt tolerant. And not naturally, artificial selection helped promote these varieties and make them genetically distinct that had this special salt tolerance. And we have increasing salinity in a lot of our arable land on the planet. And so I really saw, as I was interviewing farmers and reporting the genetic results of finding some salt tolerance genes, that our food security is really linked to the genetic diversity within species for these crops and for the crop wild relatives, where you can continue to introduce new alleles. And what happens when we lose biodiversity, whether it's in our food or it's in our landscapes outside of our gardens, is when we lose biodiversity and we have climate change, we really exacerbate environmental conflict. And so this could be where you run out of water, you no longer have crops to farm, so you can't provide for your family. So there's civil wars that break out, there's terrorist recruitment that happens. And I was at a meeting when I was a AAAS fellow after my postdoc with the Department of Defense Combatant Commands, talking about all these predictions of wars and conflict that are going to happen around the world related to climate change. And I asked what biodiversity information they use, and they said, oh, we don't use any. Lockheed Martin makes our models. They don't use any because it all correlates with infant diarrhea reports in hospitals that we can easily get the data from. Like, it? What if that massive definition of biodiversity is it? So I'm still stumped, and it bothered me. It ate away at me. And so I'm going to show you a couple of reports that are recent. And people aren't even using these models very much. Like, they're still relied on, but people don't take action. When there's an early warning, they're still not taking action. And there's still only about 80% accuracy in these early warning conflict models, even the best that our government can make. or bi. And what we need is not a Superman to react to an early warning. We actually need a sprinkler system to prevent that from happening in the first place. And again, these are some of the types of data. A lot of it is satellite-based data like temperature, precipitation, but you've got governance, GDP, all of these different elements going into our models. This would be what an example of a model would look like. And biodiversity is left out. So here's my thought that I stuck with before I met Beth and before I found these amazing opportunities at the University of California to do biodiversity research, is can biodiversity be our intelligence tool, like a strong indicator of processes that are happening on the landscape? And can it be our object of protection because it has that inherent value of 4.5 billion years of evolution and its services that it provides for us? And can it be our sprinkler system? Can it be the way that we can work together with biodiversity to take preventative action if we understand it, what it's doing, its ecological roles? If it can be any of those things, it has to be regularly observed. So we need it to be as regular as satellite data. But the satellite data on its own is not going to provide enough because the satellite's only looking from the top down. So it's only measuring the canopy. It's not getting all the juicy details of the jungle that you're trekking through and the understory. It's not going to be getting the monkey on the tree or the ferns or the lichens and mosses that this guy, or the guy, like he's not in the picture from the satellites either, even if he's wearing a red shirt. So we need to have on-the-ground measures that complement those large swath measures. And so that's this connection of scales that's such an issue. And one of the opportunities to make really regular measurements that I really believe in, that I'm so excited about to talk to you about for the rest of this lecture, is eDNA, environmental DNA. This is what's shed from organisms into water, soil, air, mucus, feces. You can find it on leaf surfaces. It's on this microphone from me, sorry. I can't help it. I'm breathing. And sometimes when you scoop up some soil, maybe there's some whole life cells, maybe there's something that died a couple weeks ago, maybe there's DNA shed from something that passed through the environment. So you kind of have this mixture of what's really local and what was in the area, or maybe, a bird pooped and what it ate from further away, the DNA ended up over here. And so it's kind of imperfect. It's semi-local in space and time. But when you pick it up, which is really simple, you just put on some gloves and you pick it up in a tube and you get it to a lab that does a bunch of fancy stuff and sequences it, you get a forensic picture that you can kind of put back together of what that environment was like a little time capsule. And it is a time capsule that's ephemeral, unless you freeze it. So it's kind of perfect because you don't want eons of DNA to always last there because you wouldn't be able to see what the season is doing, right? What's happening in that moment. But here's a photograph, a recent photograph of DNA where you can actually see the double helix. I thought you guys might want to nerd out on that. And here's the structure of DNA, and you can see that deoxyribose sugar. And then this is glucose, and don't they look kind of similar to each other? And it's also called sugar, so you get it. DNA is also food. So DNA gets chomped on by bacteria, and so it can fall apart. It can disappear. It can get eaten very easily, and so it doesn't last forever. So maybe when you're collecting soil or sediment, you're getting a signal for a couple of weeks. Maybe when you're collecting water, you're getting a signal for a couple of days. So back in the early days when the UC Conservation Genomics Consortium just started, we were all talking a lot about what we can do for California conservation, what projects people had, what ideas, what software needed to be developed, who had what samples, and sort of everyone was at the table. And here's some of the early people that really shaped this decision of what to do to help get eDNA off the ground in California. And you can see Bob Wayne here, and there's you, Beth. I just realized, and a couple of other familiar faces in the room. Mike Dawson at UC Merced, who is part of the consortium team. Brad Schaefer's there, Ian Wong. And so we decided to make a citizen science project where we would invite people to sample, collect samples, sequence them, curate the data, put it online, give it back to people to help analyze and communicate the results, and then that would cycle into the next foray of what we would invite and study next. And we would support grassroots projects so we could help amass data, make common methods so everyone could really help each other out and understand then how to troubleshoot. And then we would make these biodiversity baselines where we would start with California and we would get species lists on the way, but we would really start to see alpha and beta diversity changes, like little time capsules that we would collect that we could go back to and see, oh, were there fewer species before the fire hit? And so, or how has the composition of this landscape changed after this disturbance event? And so we keep these in minus 80-degree freezers as part of the service that CALeDNA provides for the state. And so we launched this program, and over the years we've changed a lot. We've moved from the citizen science model to sort of a community service model that I'll talk about a little bit more. But we've supported many different scientists doing these grassroots projects. And this is some of the founding team at UCLA, where it originally was headquartered. Now it's headquartered here at UCSC. And we have now over 50,000 soil, sediment, and water collections in our freezers. The sampling is, again, quite easy. This is Dora, one of our eDNA technicians, collecting sand samples. We collect in 3 biological replicates. For water, there's many different ways that we filter water. We're actually trying to hack and improve this system right now to make it more easy for collectors and more inexpensive because it's a lot of, and like less plastic use. But our sampling design is collect some water, put it on a filter, extract DNA, or collect some soil, and then from a 1/4 gram, just a little bit of soil, we're going to extract DNA. And then we do lots of PCR. So we really try to get everything across the tree of life besides viruses. Unless someone really wants to know about the viruses, then we'll talk about that. But usually it's bacteria and archaea from 16S, fungi from ITS1, invertebrates and protists from CO1, plants and green algae from ITS2, vertebrates from the 12S region, and eukaryotes from 18S. And these are barcode regions that have been, for decades, sequenced in little pieces from those 205,000 plus species that people have been surveying with DNA for decades. So there's a lot of reference data for these. And so I'm not going to go into too much detail about this, and I'm happy to follow up with people afterwards on the methodology. Also on our website, we put everything there. All of our protocols are open. So barcoding would be you sequence one single sample that's a single organism, and you get its ID from its DNA barcode, its unique DNA sequence. But metabarcoding is where you have a mixture, like the soil sample, you might have lots of species, and when you do PCR, you're amplifying all of them, and you're able to get a whole bunch of those different targets within your group, like fungi. So when you just do metagenomics and you're just sequencing all the DNA without doing all this PCR work, you're getting mostly bacteria and a little bit of the other components of the ecosystem, but you're able to really get across the tree of life when you're doing metabarcoding. So it's imperfect, it's genetically limited, but it is still a huge amount of data. Okay, so in a single sample of soil, and I recently just I picked a single sample from Alameda County, not too far away, we get usually about 400 bacteria and archaea, 200 small eukaryotes. So that's what bacteria and archaea look like. And here are smaller eukaryotes. We get rotifers, we have apicomplexans, plasmodium, the parasites are in this group. We get about 45 invertebrates and fungi, and this accidentally has sound, sorry about that. And we get about 10 plants, 30 green algae, and maybe, if we're lucky, 5 vertebrates. Usually human is one of those. Okay, so, and actually, let's go back here, because when we also think about scale, which is coming up more and more during this talk, the smaller organisms are more high in number. And this is just a body size rule of life, that if you're small, you are evenly distributed in the environment, and you're allowed to be everywhere, and you're going to be numerous and easy to find. And if you're big, you're going to be more sparsely distributed in the environment, still very important, but we're not going to see as much. So we see these declines in numbers in an expected pattern. And from these, we can calculate metrics like alpha and beta diversity, just like I showed you from the spaceborne or airborne measures. So we can calculate species richness. This is one of Jen's projects, who is the head of science for CALeDNA, even though you've given yourself a new title recently. Chief, Jen, help me out. Jen's manning the chat right now. Chief Project Scientist. Okay, great. So Jen has a project at Swanton Ranch, just up Highway 1. And it's looking at wildfire. And there's a couple of different depths of soil. And we can see that for species richness, it's almost like you can pinch the plot, where when there's a burn, there's less variation in the burned areas at depth, at mid, and at surface levels than at the unburned sites. Can everyone see that? Cool. So that's great, but it doesn't tell us why, right? It's an interesting pattern, but we need the geospatial data to help interpret it. Okay, and then here, we've got beta diversity that we can look at from eDNA, and this is LA River samples. There's about 450 in total, and the blue-green ones are concrete channel, the really urban part of the LA River, and then the soft bottom ones are the yellow to brown, and you should be able to see them partition out, but we don't know if this is the only driver of biodiversity variation along the LA River. So there might be other things as well that are contributing to these patterns. This is an ordination plot where the dots that are close together are more similar in the community that they share.
Do you know the greenness that's evenly distributed left and right?
That is mostly concrete, but with some broken up matter. So it's sort of a mostly concrete. Great observation. All right, so going back to these aims, I'm going to ask you to revisit the baseline goal for CALeDNA with the context of scale. So let's think about two kinds of scale. We've got absolute scale and relative scale. Absolute scale is where the human, or you as some reference point, is a single static reference, and you can ignore those grander ecological forces. So I'm looking at the size of this rock by relating me to this rock. And then there's relative scale, where I'm relating the mountain to this rock. And so I might think, oh, that's a big rock, but there are other bigger rocks on that mountain, or I'm getting a broader ecological context. And they're both really important. But relative scale is how you incorporate shifting baselines. And so maybe it's like, oh, you need to have the same, maybe there's seasonality, maybe there's other processes that are happening. Let's go into 3 more other lessons on scale and how we would visualize a baseline. What's our grain and extent? How big is our net? And how small is our mesh size for what we're catching in our net? What are those scales that we need to use? What about our observer position? If we're riding an elevator up and down looking at a landscape, we're going to have a different view of what that biodiversity baseline might look like. And what about space versus mass? How do we not just focus on the number of species, but also focus on the functional relationships and the openings within an ecosystem? This is from a really awesome paper from Sutton, 2018. Okay, so quick quiz for you. Which scales are we talking about here? This is current and future projections for 2080 of California rare flora hotspots. Is it absolute or is it relative? Great. I think I heard relative and I heard absolute in the audience. So it's both. We're using today as a reference, so that's absolute. Our standard spatial scale is absolute. They're both being measured by the same scale. But the dynamic context of looking at this range shift is the relative stability. Great, so now you're ready to go to the next level. We're going to look at organizational levels, where we have the microhabitat that eDNA captures that's under a rock, going all the way up to the watershed scale. These are nested in hierarchy, but the influence can go up and down. And if you're, say, a coastal lagoon, sometimes you could be influenced by things that are happening at the entire watershed above you, and sometimes maybe they're not influencing you. But the small things, what's happening in that microhabitat, we think is better measured by eDNA, and the larger scale, like watershed and valley scales, are better measured by remote sensing, mostly in their convenience. And so to kind of figure out how to put scale to connect eDNA and remote sensing, we have all these amazing people. There's more than I couldn't fit on this slide that were involved in different research projects that really helped us kind of get some proof of concepts out. And our early work was this landscape eDNA paper, where we took a bunch of remote sensing variables from what we call Earth observations as predictors to see how they predict family level information from eDNA. And we could see that, yes, we can actually, with 35% explained, it's not great, but it's not terrible, we can actually model California's biodiversity. And we can see that the habitat, the macrohabitat scale, we don't really see any separation out of eDNA. Like, it's too lumped. But the microhabitat scale, we see very distinct clustering. So eDNA is working really well at the microhabitat scale. Okay, so we keep scaling up the program. We've got momentum. We're excited that eDNA and remote sensing can be combined. We try to make eDNA work easier. So we have software. This was led by Dr. Emily Kurd to produce the sequence data to taxonomic results pretty easily. We have a lot of wet lab improvements that happened once, largely when we moved to UC Santa Cruz, because the paleogenomics lab had been optimizing all of these different assays and figuring out how much error there was in replication. And so wet lab improvements happened there. And then we had some projects. Our big grant had ended, but the National Park Service said, we want eDNA surveys in Hawaii and Alaska and in California. And Metabolic Studio, a group of artists, wanted eDNA surveys for the LA River to help connect communities and think more creatively on how it could be revitalized. So we had these projects to build and continue to support students and grassroots work. So I'm going to highlight this awesome work that was done by then graduate student Suzanne Lipton, who just recently finished her PhD. She was studying the microbes from dung beetles and relating them to carbon, and could figure out like their relationships, make hypotheses about their functions, and then validate some of that with like doing literature review of what they were doing to the soil. And we had some other grassroots projects. This is from UC Merced. They were looking at seasonality in vernal pools and seeing if the different layers of a vernal pool system that can be very small were distinct from each other with eDNA. And they found that they were. And that from one year to another, you could have different patterns. So back to scale. Now we see all these projects build up. We always have to ask, okay, for this project, for the eDNA that we think we're going to be sampling, how big is here and how long is now? And that was said first, way before we got there, by Helen and Newton Harrison, who have a huge legacy at UC Santa Cruz. So involve artists early, and they'll think faster than you as a scientist, and they'll, save you a lot of years of work. One more case study to highlight, this Woolsey Fire case study I like to talk about, because we found in these lagoons after the Woolsey Fire in Malibu that the burn sites had this whole watershed scale that they were correlated with, the alpha diversity. The unburned sites were only local eDNA, but after 1 year we got to watch it reset, where everything was local only again. So you actually just have a simple correlation factor that can tell you about disturbance, which I think is so cool. So I could sample eDNA, correlate it with different scales of remote sensing variables, and tell you how disturbed the site is. Okay, so now I'm going to go on to the biggest opportunity I think our lab has had yet, which is Bioscape. This is a NASA program that they invited people that were to collect on-the-ground measures to relate to airborne measures, where they were testing out everything that's going to go on that surface biology and geology satellite. They're going to put on planes and fly them over the Western Cape, one of the most beautiful places on Earth, with tons of endemic species and beautiful biodiversity, extreme habitats. And they liked it because there's not a lot of big canopies. It's like everything's kind of short shrubs there. So easy for their sensors, but super diverse, where you have a lot of recent evolution that splits species. So they should have only slight differences in their leaf spectra, and so that's going to challenge their sensors. So we competed, and Madeline Slemp is a graduate student in the audience who is the lead graduate student researcher for our Bioscape project, and made some slides that she recently presented, so I stole them. Thank you. But this is your work. So we pitched that, eDNA is really great for connecting with the remote sensing data. It's going to help us understand what scales we're able to see, and we're going to look at rivers. And we're going to build on all the experience that CALeDNA has in California, because it's another Mediterranean climate, just like the Western Cape. And we're going to collect water samples, macroinvertebrates, soil and sediment, and vegetation surveys at the same time to line up with the airborne data. And so we're in there, eDNA in field, and we're coordinating with all these other projects, and it's really exciting. So I'm going to show you a 2-minute video about Bioscape, and we're one of the few featured projects in it. Okay. They were flying these planes for a month straight to be able to get everyone the data that they needed. This is one of the projects that was looking at salt marshes. It's not our project, but it was still really, really cool to see what we can learn about how these plants survive in these extreme environments. Next, Phil Townsend's group from Wisconsin is collecting leaf spectra to be able to align with the remote sensing data to predict the species and their stress responses from all of these different native fynbos taxa. And then this is us. We spent 2 months surveying rivers, the Berg and the Ersti that I'm mispronouncing, collecting everything that I said we were going to collect. And then we did all of the DNA extractions in South Africa and are fully Nagoya compliant. And then this last team was looking at kelp, at the genetic diversity and chemical diversity and habitat diversity of kelp forests. It's just absolutely stunning. And then everyone was talking to each other and sharing their stories and fieldwork, and it was about 50% South African, 50% American for the collaboration networks for science here. Okay, thanks. So here's what the flights looked like, and you can see a lot of them are ours. They had to go up and down and up and down. They complained so much because, oh my gosh, there was a jet fuel shortage. They had to go to Namibia to get fuel to fly the planes, and we were trekking an entire river. A lot of people were just going to one reserve, so they had to fly a lot for us. Anyways, these are some of our sampling sites, and we sampled soil, we sampled sediment, and we sampled water, and then did those other surveys for invertebrates where we collected them and then stored them in ethanol, and we extracted DNA off the ethanol. So again, these are more of Madeline's slides. We already see with some of these remotely sensed measures like NDVI, we already see some relationships between the eDNA data and the remotely sensed variables. And then we see that there's different biodiversity in the different types of substrates that were sampled. And in collaboration with Random Forest, a machine learning modeler named Ryan Martin, who is in Canada, Madeline found that some of the different substrates like soil and water and sediment could be really well modeled for beta diversity, so that compositional similarity or differences to like over 50%, some of them 75% of the data explained in our R-squared. And the largest drivers, so these are really looking at the scale, at this phase I'm presenting the scale over time, but the scale over spatial scales we're still doing right now. So she found that bacterial communities are predicted by decadal. So if we do annual, every 3 years, decadal and 30-year windows, decadal precipitation variability. Eukaryotes are predicted by multi-decado drought extremes. This is hundreds of variables going into these models. Arthropod diversity is largely driven by human disturbance, thermal regime, and vegetation phenology. Mammal diversity, for beta diversity, is responding to vegetation productivity and long-term soil moisture variability. And plant diversity is predicted by current vegetation health within 30-year with 30-year hydrological stress and atmospheric deposition. And fungal diversity tracks current tree cover, atmospheric pollution, and decadal precipitation patterns. So yes, it's messy and complicated, but we can actually figure out these relationships here, which is exciting. And now what we're doing is we're taking the functional diversity from all the hyperspectral data that is telling us so much more, and we're seeing how it correlates when we try to predict it with these same geospatial variables. And we're going to be doing that for sites and then for sub-watershed scale, and then seeing what lessons we can apply for managers. Like, what are the sprinkler systems that we learn about? What are the early bioindicators that we learn about? Okay, so this is like our little diamond of R&D that we've gotten to work on, and we've been able to work really heavily with NASA on. But then what do? How do we move it into something that's technology? So what we want to do, what CALeDNA, the whole lab wants to do, is keep helping the world learn biodiversity with eDNA and remote sensing. And so we've really been studying what do people need, and we ended up building a web tool with some grant funding that's for the world to use, called eDNA Explorer. And then we spun it out as a company from UC Santa Cruz in order to be able to keep it alive, because it's really hard to maintain a nonprofit or a lab program compared to it being a company that can take in different, more diversified funding. And so what eDNA Explorer does, it's our partner that takes eDNA and geospatial data and AI and makes all these different biodiversity reporting for people. That's for commercial development, but also restoration and conservation. Most of the customers are restoration and conservation, and helps you with risk management and regulation. And a lot of these questions about environmental health are what eDNA Explorer is helping people tackle. And so they don't own any labs. They're working with lots of partner labs. So now CALeDNA, as like the founding partner lab, gets to coordinate with all these labs all over the world and help them get going on eDNA work too. And it gives us business in the lab for students and lab techs to collaborate with managers that are desperate to get the data. They actually want the information. And it lets us thrive and keep going into the field and doing what we love. So here's some of the people in the lab doing field surveys. A lot of this is with work with local organizations. We run workshops in Rwanda for African continent eDNA scientists early and mid-career, where we work with African parks to help guide rewilding with eDNA research. We also have one of the participants became a graduate student at UC Santa Cruz. Here's some more pictures from the field and from the lab. So we've trained now over 60 professional scientists. And we've supported student work that's been more easy to publish because they can run their projects quickly on eDNA Explorer, undergraduate research, and this is some of the team. And so people are generating data and doing their own research projects, and we're really excited to be able to grow that team. And one of the products that we recently, it actually just got published yesterday, is we made wall-to-wall predictions for the state of California of bacteria, invertebrates, and fungi that are 19 dominant communities. That's the first ever for the state to be able to include in their biodiversity assessments for 30 by 30. So it's pretty cool that the little guys are getting counted too. And not only do you get to see their distribution modeled and their uncertainty that comes with that, but some explanation of the Earth variables that underpin those community types. And so I'm not going to go into very much detail about what the eDNA Explorer offers as a partner company with UC Santa Cruz, but I'm going to give you a few highlights, and then I hope that you follow up with me or you check it out on your own. But we show people like the different species that are found in their eDNA and which ones have been locally verified. If there are any that are endangered or invasive, we help people create baselines. We help people see where organisms have been detected in larger contexts and species distribution. We help them track trends over time. We help them look at beta diversity over time as well. And then that random forest, like you saw from Madeline's bioscape work, we automate that for people so they automatically get to see, oh, ozone was a top predictor, and I actually had a pretty good performing model for my eDNA project. We automatically do that for them with hundreds of variables from Google Earth Engine. And we're integrating these data together to build health report cards for restoration projects. So you can, and this is more of that absolute scale where you have a reference site. And so I'm going to conclude with some new news. We have really exciting joint funding for projects that would not be able to be leveraged by eDNA Explorer alone or CALeDNA alone. But by our partnership, we're able to do this work that is, I'm still in awe that we're doing this. We're processing, we just finished it, I think, over 1,000 samples this year to guide restoration for River Partners, which is a major nonprofit that's buying agricultural land and converting it into functional wetlands again in Central California. We are now serving 5 districts of the California Water Boards to monitor streams for indicators of safety and species of concern. So if someone pooped in that water or there's endangered species in that water, we are the lab responsible for reporting that to the state. And I can tell you about the fecal assays. They are fascinating, but we're looking at hundreds of samples per year. And then now we just recently were funded through the Cannabis Restoration Grant Program through the California Department of Fish and Wildlife to develop an eDNA-based stream health index for the entire state, which means this year we're collecting over 2,600 samples. We have a huge citizen science effort. We got to bring it back because we have to go to 80 different watersheds and collect all of these samples between May and September when all the invertebrates are out. And if you want to know more information about it, talk to Jen because she's really the powerhouse of this project. So thank you so much. Thanks to the UCSC community. I'm really excited that we can follow up. And so I hope that you email CALeDNA or email me anytime. I hope we grow together. And I need advice on how to sort of rebrand because we're not just revolutionizing conservation in California. It's some sort of like through California helping everyone. Thanks.
Thanks, Rachel.
Thank you.
That was a great talk. There was a lot that you threw at us, and I'm sure there are a lot of exciting questions out here. Come and have a seat so you can.
Okay.
We're going to sit here. We have about 15 minutes for questions. I know there are some questions from online, but I'm going to start because I get to. And I know a lot about the stuff that you've been doing, so this is kind of an unfair question, but I'm hopefully setting you up to have a kind of fun start to this answer here. But what is something that you have discovered by sampling eDNA of different places that was unexpected and led to sort of a mystery of trying to figure out how it got there, but then was eventually solved and turned out to be super interesting, and also tells you like how easy it is to get environmental DNA into the environment.
The giraffe? Yes, are you talking about the freaking giraffe?
I am, yes.
I have been plagued by this giraffe like 3 times, actually. I don't know if you know all of the giraffe instances that we've encountered in the lab. Okay, so we're on the Russian River. This is the day before shutdown starts. So we're doing a BioBlitz on the Russian River, collecting 170 samples in 2 days. And one of the sites on the Russian River is called Mark West Creek, I think. And anyways, we find a ton, 100% match of giraffe DNA. And yeah, anyone know?
Yeah, it's the wild animal park in Santa Rosa, I'll bet.
Yeah, Safari West. Yeah. I had no idea, and I was going out of my mind. And we have a lot of the theory on how far DNA travels through rivers is from Europe. And so they were like, oh, it can travel up to 1 kilometer, but it was 2.5 kilometers downstream. So that gave us some idea of the scale that we were measuring.
Yeah, really impressive. All right, sorry.
Thanks.
Yeah, I do have a question.
I'm from LA. I've never been to that safari.
The question I had is, we just came back in November from visiting that part of South Africa, your whole spot where the NASA planes were flying. And we went to several, I think 3 of the 11 botanical gardens they have. And one of the things that surprised me the most is how many plants in your average California garden actually are South African. I knew I had 1 or 2, but there were a huge percentage. I kept going, I have that, I have that, I have that, I have that.
Yep.
And I was wondering, you sort of broke this with the giraffe DNA here, is that how much, considering that people have consciously moved this stuff over here, how much does that mess up the data that you're collecting when you go bird of paradise, plumbago, I could go on and on, there's a long list. And what that does with messing up your data. That was really the question.
That's so great. I just planted CALeDNA, which is a really great plant to put, South African species to put in gin and other cocktails in my garden in California, and I see it all over the place. I almost put it in this one tonight. But yeah, so we move species all over. That's part of our roles as humans, and it's like we're doing one great experiment. And as long as those observations are getting recorded and put into GBIF, we expect them. And so we actually filter very few species out when we say this is unexpected. It has to be like endemic in China, and you would never see it here. But as long as you're using iNaturalist and taking photographs, and some of those are domestic species or cultivated species, we can handle it. Compost, though, oh my goodness. We started, when we first were testing CALeDNA protocols out for soil sampling, we used a botanical garden because we had labeled plants, but nothing came back looking anything like the planted area. It was all random other plants from this mulch that was purchased from who knows where, getting this other species mixture. So yes, it does make things messy.
Never thought of compost. Right.
I mean, you're such a wonderful speaker. I came in here not knowing what the E in eDNA stood for.
And you still came to the lecture.
I feel educated and so excited. I was wondering, in the sense you introduced biodiversity at the beginning of the lecture, is it correct to think of it as an opposite of monoculture? And if so, that is the cash crop is the ticket, farmer's ticket to making high margins. Is there a high-level observation that broadly could go out to farmers in the world that is grounded in your science? What sort of things?
Thank you for that question. We've seen a lot of like have your cake and eat it too examples coming out lately, where you can have your higher biodiversity, so you get your biodiversity credits. If you're a large company farming, grapes, you can have more biodiversity, and you don't have to sacrifice. You don't have to have the losses. That there is biodiversity that can also increase your productivity or buffer you from disease or drought. And so there are so many beneficial species out there that you can encourage. And so if you have that fine grain scale detail, you can get there. And so there's a lot of initiatives that are out kind of ahead of the data, like not biodynamic, the other word, more agroecological practices that we still need to characterize what is it that's actually changing about the biodiversity that has these co-benefits. Yeah, so we'd love to work more in the ag space on this, and I think we're situated in the perfect place, especially being an AES for UC Santa Cruz. And we have one pilot project with the Agroecology Center here.
Let's take one question from our online audience. Jen?
Yeah, Paul Sherman asks, or he says, very important, useful, and extremely well presented, and wonders about how you're going to prevent malicious use and how to prevent other sorts of issues with sorts of exposing sites, and wonders if eDNA Explorer will take on sponsors.
Cool. Thank you. thank you, Paul Sherman. So yes, there are standards for protecting the locations of species that are threatened and endangered. And so we follow those standards even more strictly than iNaturalist does. They'll put like a 30-kilometer buffer around it. We just won't show you the locations. We'll just say, in California, this was found. It actually came up, there was a really only from one location known mushroom that was detected in an eDNA sample that someone had collected from a whole different part of California, and the whole fungus community, and they are really, really determined to find this mushroom, was like yelling at me, please give us the location. I said, I can't. I've protected myself from seeing it. But after years, we eventually hacked our system. We gave them the location. We're writing a paper on it. So yes, we've protected locations for threatened species, and we're working actually really closely with Canadian tribes right now, and a little bit with US tribes, to look at the sovereignty standards for data sharing and access that they want, and then we're building that into eDNA Explorer, thinking if it works for them, it's gonna work for the world. Where maybe you're happy to share the species locations, but not the timing of when your anadromous fish are coming through your water system. So we are really trying to be good on this, and we are aware that if you have an endangered species somewhere where someone doesn't want it because it has different policy implications, people could shoot it. So we definitely don't want to allow malicious behavior, but we do want to encourage open sharing as much as possible responsibly. Taking sponsors, let's follow up on that. I don't really know what that would look like, but it sounds cool.
Thank you. We know the Harrisons, or knew Helen anyway, and Mike. One of the things that they were very focused on was the effect of climate change, and especially in sensitive places, as you probably know, like the Himalayas, right, where just the rising of the loss of glaciers meant lack of water and messing up the whole aquasphere. And one of the things they wanted to do in California was to see if we could prototype remediation. And so I'm curious whether that is also part of your mission, to envision what is the remediation possible in a changing climate world, and what can you share about that?
We need artist collaborators, like a lot of them. There's
Explain the whole context.
Yeah, because I wouldn't call it art, but it's just that we once had a 2-hour argument about whether it was art or not.
Oh, I won't get into that then. I've certainly been schooled by the Harrisons. The mission of remediation.
Testing, can you make this?
Yeah, we don't see enough projects like that, and I think all of us in CALeDNA have observed it's like descriptive study after descriptive study. We need experiments, and there's very little effort there. We need to set up mesocosm experiments. It wouldn't take that much work to do, but it needs the creative brain power to design it. And yeah, we really need to validate a lot of the relationships that we see, especially biotic interactions. We see co-occurrence, but we can't prove that they're interacting. And that limits us in our power to communicate the results too.
I think also there's been a shift, maybe more recently, in the conservation community away from thinking about fixing things or restoration and remediation to think about adaptation. So how can we learn about what make communities more robust and resilient in a changing climate, and use that information to create communities that are more robust and resilient in whatever the future climate is going to be. So I think that's also something that I think even Josh Harrison is trying to push forward now with the stuff that their son, right? I don't know.
We've been, he's going to be an advisor on the CDFW project, actually.
Oh, really? Cool.
And a lot of this is looking at, different forms of remediation. What does that mean? Health has these different definitions depending on who you talk to, and just being more inclusive of the strategies that people take and how to design projects so that you can actually track, is it remediation where we have to expect change in 30 years? What's our time scale of measuring the effects of our efforts. And a lot of projects like the River Partners, they're doing inoculations to try to catalyze remediation of the soil. So how can we collect data that complements what they're doing?
Maybe another question from our online audience here, Jen?
Okay, yeah, this is kind of a conglomerate, but sort of general questions about how eDNA Explorer was funded and what were the advantages of moving to a community service model, from a community model to a service model? Were there more opportunities? And what's next for CALeDNA?
All of it. Go.
Come to our lab and hang out, and we can talk about that over lunch. eDNA Explorer was initially funded by an anonymous foundation, and then they ended up being part of an investment team, and we did spin out to become a company. And we're really targeting to be a public benefit corporation, so trying to make sure that people get as much free service as possible so that the community college doesn't have to pay. We are currently also very heavily grant funded to sustain ourselves as we're growing. We do have paying customers, but we have a grant from Genome BC where we're launching eDNA Explorer Canada. And so as we're standing that up with a collaboration with the University of Victoria being a partner lab, and iTrackDNA, that there's a big community of eDNA people in Canada. We're really reliant on grants to keep us building out the architecture. Future for CALeDNA and why we moved to the service model is instead of us going to people saying, do you want eDNA data? People were desperate for eDNA data, and they still are. There are very few commercial labs. There are very few academic labs that produce eDNA data, and we need more of them if we're going to make this a really standard, cheap, and easy to get measure. Yeah, it was just like we didn't have to do the work. And I think we have a really interesting funding model where students are directly paired with managers that actually want the data, and they work with them really deeply collaboratively. So I'm happy with it. I don't think it's just service that they're getting out of it.
And if we go way, way back in time, to the very beginning, the conservation genomics project from which it was born was actually funded by a multi-campus initiative by the University of California Office of the President.
The CALeDNA program.
The CALeDNA program. And then we got a little bit of funding from the state, but we also had an injection of funds from the Websters we were very fortunate to receive, which helped us train a whole bunch of students who went out in the field, including some of the people that were in your slideshow here. So we
and
are
the Swanton Ranch
grateful
project.
For the Swanton project. Very grateful for this early funding that we got, both from benefactors and from the University of California.
Yeah, and actually the National Park Service, being able to say, oh, this is a place where people are collecting a lot of eDNA samples. There is no national environmental sample collection, but knowing that Santa Cruz had a lot, which was funded from the Webster's donation, that helped bring in other people saying, we want our samples housed here. National Park Service is happy to let our samples be kept at UC Santa Cruz.
All right, well, thank you very much for all of this, and we're going to get closed out here, so...
Thank you.
Thank you very much,
Thanks, George.
Beth, for that fascinating talk. And thank you all for coming tonight. Please join us for our next lecture on March 4th, which will be about unmasking cancer's genetic code. Thank you all, and good evening.